So welcome everyone to our last seminar of this year. Today we have Mark Anthony that is going to talk us about this cool topic of soil biodiversity.
And our seminar series still has some slots for next year. And it's really, everybody can invite. One slot only left. Oh, we're doing the January one probably.
No, in April or May. Okay. Only one left.
But yeah, I mean, the slots can be filled by you guys, right? So please feel free to invite people.
Wait, you could turn that light off. Maybe it would look a little bit nicer on here.
There, behind you.
Yeah.
I don't know.
Maybe all lights would just look better.
Okay.
I have no clue. No, it's really there. I think now
It's a dimmer, yeah.
Oh yeah, that looks good. My hero. Hi everybody. Nice to see a lot of faces that I know, and it's still wild that you can be at WSL for so long and still see a lot of new faces. Hello to folks online. Today I'm going to talk to you about a couple of my research projects that kind of bridge the worlds of quantifying soil biodiversity at global scales with a more European context. What is the diversity of European forest soils in terms of microbes that are present, and what might that matter for ecosystem function? So before I start off, I just want to thank folks that have helped a lot with this work, especially Colin Averill, with whom I've collaborated a lot, who now has a really cool nonprofit called Fungal that people should check out, as well as a whole bunch of collaborators who work with the ICP Forest Network, Leo Tedersu from University of Tartu, and Arthur Gessler, of course, who's just been an amazing host for my Ambizione here at ASL. And of course, the Swiss National Science Foundation for funding all of this work.
You have to share your screen.
Oh, yeah, that makes sense, doesn't it? Good catch. That would have been so awkward if we were halfway through and nobody saw anything. Okay. Hide that. So that aside, let's start. I'm going to be talking a lot about biodiversity today. And I feel like when we hear that word, this type of picture emerges, some ethereal landscape with a lot of big organisms. And I'm not going to be talking about this element of biodiversity at all. So we're going to scrap this narrative, but we're going to draw from this logic to frame the fact that we are experiencing really abnormal changes in biodiversity. We are seeing higher extinction rates than background levels. We are also seeing that the impacts of human are abnormally rapid and expanding. And humans, we as creatures, are abnormal, and that our imprint is different from other organisms because we're conscious and we can make choices that reflect on our observations of biodiversity loss. And all of this is strongly suggesting that we are amidst the 6th mass extinction. There's a lot of awareness of this topic too. You see it in the news globally. We see quite a lot of Swiss context of this because as a country, Switzerland is actually doing a really good job at monitoring biodiversity and biodiversity loss. But one thing that we can kind of generally see from this assemblage of photos is that the organisms that are generally centered in the biodiversity discussion are fish, large mammals, birds. They're large organisms that live above ground, and that's not because they're not important. They are important, but they also do something different. They touch on our human emotions in a unique way that a lot of other organisms have not. And therefore, they tend to be the charismatic creatures that we see when we're thinking about biodiversity loss. And there's a massive bias here that's not just external. It's not just from public perception. It's also scientifically informed. The figure you see on the left here are estimates of 5 different groups of organisms. So we can see that some are really diverse, like insects. Some are comparatively not diverse, like amphibians, birds, and mammals. If we look at the proportion of these same groups that are in the red list, this is the largest list that's trying to catalog threatened and endangered species, we begin to see the mismatch. So this is the proportion of species that have been described in the red list relative to those that are described. And we can see that this is negatively correlated. The most species-rich groups like insects and mollusks are barely on the radar of these really important conservation listings. whereas species that have very few organisms in them or species in them, like amphibians, birds, and mammals, are really well represented in these inventories. Of course, we can see them. That's a big reason. We can see a lot of amphibians, birds, and mammals, and we have a harder time seeing these smaller organisms. Some people think about this as, well, it's just like finding a needle in a haystack. That's the problem. It's hard to inventory them, but that's not true anymore. It's a bias, an explicit set of biases that we have in conservation and restoration research. And I've been really attracted to understanding where these biases are. And from my perspective, a big one is soil. There are so many reasons to think that soil is a massive reservoir of biological diversity. There's a lot of soil on the planet, so it's just a lot of place for organisms to live. Soils also don't have the same type of movement that rivers, oceans, and lakes have, so there's a lot of opportunity for geographic isolation, and they're incredibly heterogeneous. So from an evolutionary perspective and from an ecological perspective, there's a lot of reason to expect that biodiversity should be very high in soil. And so what would we expect to see in soil? Honestly, we would expect to see the entire range of life in soil for the most part. We can expect to see a lot of different animals, be those small things or rather large things like earthworms. We can expect to see bacteria that might be hidden from our eye because they live inside the root systems of plants. Also mycorrhizal fungi and different types of fungi, like these more visible ectos and these more invisible or buscular mycorrhizal fungi, as well as many things that are way smaller than this, like huge diverse communities of phage and other types of viruses. So with this expectation, I set out to try and quantify how many species live on Earth and what proportion and numbers of these species are found in soil. And I did this because we already know that soil biodiversity matters a lot, because these organisms drive nutrient release. They're essential in agriculture, so for food and fiber production. A lot of these organisms are also responsible for building organic matter in soil and for cleaning water as it moves through soil into, say, the water that we rely on for drinking. This is also essential for human health. We have sourced so many of our antibiotics from soil, and many pathogens that are problematic to humans also live in soil. And of course, because of their roles as decomposers and because their dead bodies make organic matter, they're also essential for our climate system because they strongly regulate biogeochemical cycles. It's pretty safe to say that without soil biodiversity, the world as we know it would not exist. And so the first thing I want to talk about is how many species live in soil. And maybe some people know, but is anybody willing to guess how many species live in soil? Just yell it out. I won't even make you raise your hand.
10,000.
10,000. Nice.
Million.
Million.
5 million.
The stars.
100 million.
More than stars, I like that. The number is absurdly large. So it's like 10.4 billion. But that number is also super biased because viruses are freaking diverse. And we can argue the day off about what a species means for a virus. But there are ways that we can sort viruses into categories that are semblant of species. And so the vast majority is phage. So viruses that infect bacteria. But still, the leftovers still represent some estimate of around 500 million species. This is incredibly high. It's higher than most of the estimates that folks put out there, aside from maybe the number of stars there are. I don't know that answer. And if we put this in the context of the proportion of species that are there, our best estimate right now is that about 59% of species, plus or minus 15%, live or at least are in strong association to complete part of their life cycle in soil. This is a really big number. When I set out to answer this question, I didn't necessarily expect it to be this big, but what this ultimately means is that soil is the single most biodiverse system on Earth. Even within that range of error, this would position soil as being such an important reservoir of biological diversity. And so this raises a lot of important questions, like why there is such a lack of attention to the organisms that live in soil. So I looked at this week. This is the kind of summary of the red list as it stands. And most of the organisms that are on this list are creatures that are not found in soil. or creatures that don't even need soil to complete part of their life cycle. And this, to me, is quite a problem because we're overlooking probably the most biodiverse habitat on Earth. If we kind of think about this diversity based on the main organisms that we find in soil, there are some reasons why that I kind of touched at in the beginning of this. So some of the organisms that are most specialized to soil, like these clear worms, fungi, and bacteria, which have the majority of species found in soil, are sort of tricky to identify based on current strategies of cataloging organisms for the red list, which largely rely on observation by the eye. While this works for some of these organisms, like it works for the clear worms because there's so few species of them and they are visible by eye, it doesn't work for a lot of other organisms. And that's especially true for things like fungi and bacteria, the most of which are invisible to us. So with the current frameworks we have for conservation and restoration, it's inherent that these types of organisms will be ignored. And this is a huge problem. This is just one example. We estimate that there's about 6.2 million fungal species based on DNA sequencing, but the red list only has about 1,000 fungal species on here. And I wish I could say that was because fungi are incredibly resistant to environmental change and the effects of humans, but maybe they're pretty robust. They are, but they're not that robust. So this is not a scientific reason. This is an omission of our investigation. It's a failure for us to create frameworks that actually reliably work to try and catalog fungal biodiversity loss. And so I can kind of think of this effort as like a census. You take a census of a country so that you can understand where people live and hopefully in an equitable world distribute resources proportionally where people should get them the most. I can think of this as a type of census that really shows that we're out of balance right now and that more resources, effort, and understanding need to flow towards understanding soil biodiversity. And I think there's 4 key things that we need to see happen. One is new monitoring programs that need to specifically focus on eDNA as a detection tool. These are starting to exist now, but we need to incorporate these into a lot more places. And we specifically need to fund this in the global south so that this is not just a catalog of the global north. We also need to protect hotspots of soil biological diversity. Even if this looks totally different from what we would see above ground, maybe we're gonna have hotspots of soil biodiversity in an ugly farm field that's been abandoned. And it'll be totally ugly to us. It will not support organisms above ground that we care about, but that doesn't mean we can't necessarily put that as a place of protection. We also need to think about new restoration tools because most of the programs and strategies we have don't think about soil. For example, one of those could look like programs that are actively restoring small mammals that are important for the dispersal of soil organisms. And I think most importantly, we need funding and attention, and this puts a lot of responsibility on scientists to talk about this issue and to communicate it beyond just our own research bubbles. And so for me, this creates a big goal where I want to quantify soil biodiversity and understand its importance for ecosystem function. Because as much as I can look at a petri dish of fungus and have empathy for it and think it's cute and like it, most organisms can't. And it certainly can't compete with a panda. So we need to make a case for soil biodiversity. And I think part of the way we can do that is understanding just how important it is, quantifying that importance and talking about it. So for the second half of this presentation, I'm going to zoom us in to a slightly smaller spatial scale, so the scale of Europe, and think about what is the soil biodiversity here from a microbial perspective. So really just focusing on fungi and prokaryotes, and what might structure these communities, and ultimately what might the importance of this be for carbon storage. I'm focusing on carbon storage because my work takes place in forest ecosystems. And although forests do a lot of things, one really important thing they do is store a lot of carbon. So this is where this work has been conducted. We collected a bunch of soil samples from across the ICP forest network. It's not very important that you know what that is, but it is a set of harmonized forest inventory programs. We collected soils from over 18 countries that were forests dominated by either broadleaf trees or conifer trees. And you can kind of see the mix of those here. Switzerland's well represented on this map. And the first thing we tried to do is quantify how many species of fungi and bacteria live in this landscape. Because this is maybe the highest resolution inventory in forests, we could use some statistical tools to approximate how many species we would expect to find and which species and lineages are most common. So across Europe, we estimate a surprisingly small number of fungal species. I expect this to be a lot higher, but we approximate that there are about 14,000 fungal species in forest soils. These include the organisms that we would most expect. So these are largely represented by the Basidiomycota and the Ascomycota. These are later evolving fungal lineages of the Dicaria, and they include some of the most important organisms that form biotrophic interactions with plants. And I'll talk a lot more about that later. In terms of the genera that's most common, this is Russula, the brittlecaps. They represented about 10% of all of the sequences. So there's a lot of variation here. This is a huge mean, as you can imagine, but still, this is cool. Russula is a really interesting and diverse lineages, so I'm not super surprised to see this. But the fact that they as a single genera can represent 10% of a community is pretty impressive to me. In terms of prokaryotes, we're looking mostly at bacteria because the molecular techniques we used are not as amazing for archaea. Here we estimate that there's between 1 and 2 million species. I don't want to go into the details of why there's such a big range, but there's quite a discussion about how to define what a species is from a molecular barcode, specifically as it comes to bacteria. And so that's why we think somewhere between 1 and 2 million species. If we use the more traditional strategy, we would sit around 1 million. And this again includes many of the lineages we would expect that are common in smaller scale forest inventories, particularly the Parobacteria. And then these are sort of new names of bacteria that are a little bit confusing that we're all trying to keep up with now. Some of the more traditional ones are like the Proteobacteria, Actinobacteria, and the Acetobacteria. The most common genus of bacteria were the Rhodoplanus that captured about 6%. And these are really cool creatures. And I think one kind of fascinating fact to me from this is that if we consider fungi together, both Russula and the Rhodoplanus use sunlight for energy for the most part. And so of the organisms that live in soil, you'd think that they would use soil carbon, but no, they get most of their energy from sunlight. So I just talked about one dimension of biodiversity. I talked about gamma diversity. This is the total amount of species that are in a very large landscape. But there are two other really important components to biodiversity. That's alpha diversity, so the number of actual species you see at a particular site, as well as beta diversity. Beta diversity characterizes the species composition, and therefore it's a useful metric if you're comparing between sites. Just as like a very simple example, we could think of this as site 1, which has 2 species. One of these species is shared with site 2, and so their beta diversity is about 50% equivalent. If you compare site 1 and site 2 to site 3, where there's no species overlap, their similarity would be 0%. So what I'm going to show next are a series of graphs that kind of summarize this idea of beta diversity. And so you can imagine these points as representing a particular site that are further away from each other being the most dissimilar to each other. So on the left-hand side here, we have fungi. And on the right-hand side, we have bacteria. The arrows are showing different aspects of the environment that are correlated with these community types. And one thing that I think is just visually very apparent is if we look at fungi, you generally see this clustering into the diagonal corners of this figure, where the conifers and the broadleafs are quite distinct from each other. If we look at bacteria, you see somewhat of a similar pattern, but there's a lot more mixing here. And so bacteria are less different based on what type of tree is there compared to fungi. There are other environmental factors that can capture some of the dissimilarity we see in the communities. That is the beta diversity. That is what is responsible for the different types of species we see there. And I think an easier way of showing this is the correlation between beta diversity and these different environmental predictors. And so on the left-hand side here, we have fungi. On the right-hand side, we have prokaryotes. And kind of just as you can see in the ordination, the top 3 environmental factors we have are 3 really important forest attributes: how quickly the forest grows, how old it is, and what type of tree is dominant there. And a lot more variation is explained for fungi with these attributes than there are for bacteria. So fungi are just generally better bioindicators of the vegetative component of forests than bacteria. Bacteria have more variation explained by soil factors that are not altogether surprising. For example, soil pH. It's been known for almost 2 decades now that soil pH is one of the largest driving factors of bacteria, which is important because it shows that we're getting at something that is consistent with previous analyses so that anything new we find might be credible on some level. For example, this difference between the vegetation characteristics between fungi and bacteria. So that kind of describes what diversity looks like across Europe. And in this final part, I want to talk about how the soil microbiome itself may predict some important forest carbon storage metrics. I'm going to focus on the two most significant pools of carbon that get stored in forests. The first one is soil, and the second one is living vegetation. And because I don't have a ton of time, I'm going to focus most attention on the links to living vegetation. So living vegetation is characterized by how much carbon is there and the rate at which it accumulates. And the way we can quantify this is with standing tree biomass carbon stocks and with tree growth rates, with tree growth rates representing the amount of carbon that accumulates and the biomass carbon stocks being the amount that's actually ultimately stored in a forest. And what we can see here, when we control for other very important variables that predict tree growth, such as nitrogen deposition, mean annual temperature, forest age, tree density, other things that I don't need to make a laundry list of, but trust me, there's a good amount in there. We still see this really strong signal of both fungal beta diversity, which you can see in the first column here. This is just a kind of unitless axis. There's nothing better or worse about it if you're negative or positive, you're just different. So if this is a negative correlation, it doesn't really mean anything. It could be a positive correlation, it's just difference. And so what we can see is that the difference in the fungal communities with both the stock of carbon that's there and the rate of accumulation is quite strongly linked to the fungal community. And interestingly, if we look at the richness, so the number of species observed at each site, this is just the amount we observed, it's not extrapolated as I did before, just what we observed at that particular site, it's positively correlated with these two vegetation outputs. And just like we might expect from the earlier analyses, when we look at bacteria, those that just flew up here, we see no pattern at all. So the composition of bacteria is not linked to the stock of carbon there or its rate of accumulation, nor are the richness levels. And fungi themselves are the organisms I study the most. So it's sort of hard for me to say fungi do, because not all fungi are alike, they're super diverse. And one way that we have traditionally thought about fungi is with their ecological strategies and their trophic modes. So a lot of fungi that are represented there are pathogens, like this Cryphonectria parasitica. It's quite charismatic, it's also adorable, so it's so shameful that it's so destructive in forests. We all know what pathogens do. Pathogens infect us. They're not a new group of organisms that we have to understand, but they're also saprotrophs. Saprotrophs do not infect living organisms, but rather they decay dead things, and that's how they obtain their energy. There's a lot of variation within the category of saprotrophs, but for the purposes of today, I'm just going to clump them all together into a single group. We have mycorrhizal fungi and endophytes. These are two biotrophic groups with plants. A lot of the mycorrhizal fungi include ectomycorrhizal fungi, which will be the focus of my talk today. Most of the mushrooms that you might hunt for and see in the forest are mycorrhizal fungi. These are fungi that live in the root systems of most tree species that we observe in Europe, and they are part of a symbiosis that is really characterized as a nutrient exchange. So mycorrhizal fungi are better at getting nutrients, but they rely on plants for carbon. And so in exchange for carbon from the plant, they provide plants with nutrients. They also provide a bunch of other services, but that's sort of the main one that mycorrhizal fungi do. Endophytes themselves, okay. Endophytes are frozen. One second. We're back, okay. Endophytes are also a biotrophic group of fungi. They have a lot of flexibility, so sometimes an endophyte can be a little pesky and act like a pathogen. Other times it can be very beneficial and it can be a bona fide mutualist. Endophytes also use plant carbon as a primary energy source, and they can provide a whole range of services to plants. They can induce hormone production that can cause plants to grow more. They can also literally produce the same hormones, and therefore the plant doesn't need to invest in them. They can take up nutrients and act very similarly to mycorrhizal fungi. So if we separate our observations out across these different groups, we can begin to understand why we might observe the pattern we previously saw. So what we're seeing here on the x-axis is basically an effect size. This is our models that we use to predict tree growth while accounting for all covariables and what's left over for the fungal effect that's in here. On the top, we see the total fungal effect, which I showed you earlier. And if we're looking at just composition right now, so beta diversity, as I showed you before, we can see that these tree biotrophic symbionts really emerge here. We can see it with endophytes and ectomycorrhizal fungi. I put ectomycorrhizal fungi into two different categories. It's not particularly important that you understand why, but we see similar patterns between the two of them. And this is really important because the effect size of ectomycorrhizal fungi was higher than all of the other groups when they were examined individually. And this is now the second time that I've observed this in my research. So in our work last year in ISME, we found that if we look just at root tips, so if you go next to a tree, take a soil sample, pick all of the root tips out in a harrowing effort, and then sequence those, that the composition or beta diversity of ectomycorrhizal fungi was strongly linked to variation in tree growth rates across Europe. And now when we do it differently, we just look at bulk soil, so that excludes roots. This is just the species that live in soil. We find a weaker effect size, but the same pattern, where the composition and dissimilarity of beta diversity in these communities explains quite a good amount of variation in tree growth that's not captured by other factors that predict tree growth. I'm not trying to say this is causal here, just saying that they're linked. And we'll talk a little bit more about that later. One thing we do know, though, is that if we experimentally manipulate communities of ectomycorrhizal fungi and inoculate them onto trees, they do have causal effects on tree growth. So it's not as though ectomycorrhizal fungi are simply responding to what trees do. They drive what trees do. So any type of argument that ectomycorrhizal fungi simply respond to trees is just simply inaccurate. We know that they cause variation in tree growth rate, which is tantalizing to think about when you see an observation like the one I had before. If we think about species richness now, so same figure, but if we look just at the white figures, the most important group here were endophytes. And in fact, the richness of ectomycorrhizal fungi is completely unlinked to tree growth rate. This is a visualization of what that looks like. So we have tree growth on the y-axis, endophyte richness on the x-axis, and we see this nice positive correlation. This is super interesting because we've never observed this before with endophytes. They have received a lot of attention in the biotechnology sector, but mostly just out of ease of what happened to be cultivated, not because of any type of community ecological factors of them. And so if there actually is something to this phenomena, if endophytes can have some type of causal effect that's additive based on the number of species that are there, that could actually be pretty dang useful, especially because there's literally thousands of patents out there already right now for endophytes that are already being widely used in a lot of places. If you're interested in this, I'd take a look at what they're doing in New Zealand with endophytes, for example. They're largely deployed across the landscape already as a form of biotechnology. And we also know from research that a specific group of endophytes, those that you would expect to see in a soil sample, which are the dark septate endophytes, these are fungi that live in roots. You can often find the same communities in roots and in soil. And they also, like mycorrhizal fungi, will live in a root. And some of them, not all, will produce lots of hyphae that goes into the soil, which suggests that when you look at them in soil, that these species could exist, not in a state of saprotrophy, because we are looking at soil without roots, but rather as an extension of an active symbiosis. These have really large general positive effects on plant growth. This is a kind of old meta-analysis at this point, but it shows really strong effects of inoculation with dark septate endophytes on plant growth. There's a lot of variation here, and that's because which species that are present has a hugely determining effect on the growth response that's observed, which would suggest that the biological diversity of endophytes matter for tree growth. So I just talked a lot about living vegetation, and we don't have a ton of time left. I'm not going to spend a lot of time on the soil results that I have, but I just want to put this into a short context. So if we look across Europe, the relationship between soil carbon storage and the mineral horizon. So if you go out to a forest, you rip out the forest floor, that like upper brownie, and you look at what's beneath that, where most carbon is stored in soil. There's quite a nice positive relationship between how much carbon is in soil and the standing stock of biomass and the rate of carbon accumulation above ground via tree growth. This isn't fully surprising. I mean, this is what a lot of Earth system models rely on, this correlation, but you don't see this everywhere. Sometimes soils can saturate potentially, and therefore you don't see this positive relationship. In some ecosystems, you also see that when the vegetation growth is going up because of a strong priming effect, that is, the amount of carbon entering fuels microbial metabolism, you also get this big flush of carbon loss from the soil. So you can observe a negative correlation between these two. So it's actually really important that we understand that in Europe, these two really important pools of carbon are positively correlated. What this also means is that anything that's correlated with tree growth and tree biomass carbon stock is then indirectly linked to soil carbon storage. And so when we looked at the variation in the microbial community, there was really nothing with mineral carbon stocks. So microbiome richness, microbiome composition of both fungi and bacteria was basically totally unlinked to this y-axis that we see here. There was a really small effect for bacteria, but the effect size was perhaps not ecologically meaningful. So if we then think about the results we saw with fungi, the link between tree biomass and tree growth, this means that fungi are not just indicative of tree growth, but also soil carbon storage, albeit indirectly, which makes the composition and richness of fungi a very unique biological indicator of total forest carbon storage. Admittedly, we did not look at fallen wood and other things that are important, but the vast majority of carbon. So we can kind of round up to say total forest carbon storage. And if we think about what that might look like, forests that store a lot of carbon, both above and below ground, have high endophyte diversity. I didn't show you the unique species because I did not want to bore everybody to death, but they also have very unique species of trichoderma and russula species. The types of forests in Europe that have the least amount of carbon stored above and below ground have low endophyte diversity and unique species of Cortinarius and Inocybe. And again, I'm not going to bore you by going into the ecology of these fungi. Just know that this is cool. And I would therefore argue that there is actually no other group of life that is so indicative of forest carbon storage, not even individual tree species. And to me, this is a really big reason to do a better job monitoring, conserving, and restoring fungal communities in Europe. I want to squarely address a cause-effect conundrum here. I am not saying that the observed correlations here are causal. They might have some causal effect here, but I can't disentangle this based on an observational study. So really what we can say here is that there are strong bioindicators, and I think that should be the focus. We are trying to disentangle this. And if anybody has an idea for how you could literally manipulate an entire forest fungal community in a realistic way, I'm all ears, but I've yet to be creative enough to figure out exactly how you could do that. So we've had to use other ways to try and get at cause-effect relationships based on our observations. So one thing we did was collect a bunch of soil from 6 sites with incredibly different ectomycorrhizal community compositions. So this key group that we know is different, that can capture a lot of unexplained variation in tree growth rates. Then we conducted a mesocosm experiment. We focused on Scots pine sites just to kind of keep things a little bit homogeneous here. And in half of them, I don't want people to look at this yet, so I'm going to go back really quick. In half of them, we sterilized the soil that we used when we inoculated it into a common growth medium. Just imagine like a sandy potting soil mix, and you add a little bit of sterilized field soil, a quite small amount, so you wouldn't expect the physical and chemical properties to differ. Then in the other half of them, we didn't sterilize it, and we put that in. So you can kind of then see whether the biological or the chemical components of the soil inoculation could cause variation in tree growth rate when literally everything else is held exactly the same. And so if we look at this top row, here we have plant productivity and the sterilized soil inoculations across the different sites. So what we see on the x-axis are the different ICP forest sites. The numbers are not particularly important, but Switzerland's well represented in here. We see no differences. They're all the same. There's no significant differences there. But when we inoculated them with living soil inoculation, you see this incredible range in the productivity of the trees. We also did this under ambient and elevated CO2 conditions, and you can also see that the magnitude of the plant's response to elevated CO2 strongly varied depending on which soil microbes were there. So this itself shows that the soil biological community does have a causal effect on tree productivity when you hold everything else constant, kind of like we would have expected to observe based on our observations in the field. Again, the soil is super diverse, so we don't know what component of the soil is driving this. All we can do is attempt to characterize it and see what's correlated. And one thing that is somewhat decently correlated is fungal composition. So here we're showing the difference between the living and the sterilized. So it would be how much more did this plant grow in comparison to soils from the same site under the same atmospheric conditions, but inoculated. So anything positive means it grew more with inoculation, negative means it grew less. Here we're just looking at the root to shoot ratio as just one example, which suggests that the fungal component of the soil community might at least have some causal effect on differences in growth rates that are observed in this microcosm experiment. If we scale up a tiny bit based on a meta-analysis we did, here we were looking at if you add a small amount of soil from a nearby intact ecosystem, primarily in forest systems, to a pit of soil that you just planted a new plant in, how much more does that plant grow? And you can see here the effect size that was observed in terms of the biomass increase in the plant. On average, adding soil from a wildness area into the pit before you planted the vegetation out increased growth by 64%. You can see the distribution of observations on the top here in the histogram and in the points that are here as well. And there's obviously a massive range in the outcomes that you observe, which when I look at, I don't know the answer to. I have to imagine at least is in some part due to variation in the microbes that were sourced from that wild neighborhood before they were added. So this also quite clearly shows that soil diversity can steer plant growth. And that doesn't just have to be in the case of when you have perfectly controlled conditions. It can also take place outside when environmental variation is massive and large. And of course, that environmental variation in the background is also steering what we observe here. And so microbial biodiversity variation definitely does cause dissimilarity in plant growth. It's just a matter of connecting this observation experimental work, which is technically hard, possibly impossible to do. And so I think next time you go out to a forest, you may be able to see a sporocarp of a fungus. And if you have your ID book, you might be able to look it up. And if you know something about that fungus, you actually can then make a pretty informed prediction of the carbon storage at that forest, at least in Europe. So I want to thank you for your attention. I just want to thank everybody at WSL because I'm leaving in like a week and a half now. It's such a cool place, and I've had such an amazing time here. It's such a special environment to grow as a scientist, so super honored to have gotten to spend time here. And if anybody has any questions, I'm happy to take them.
A little comment.
Yeah, please.
Okay, so about phages. So because we are studying the viruses of fungi, and our last work is of soil-borne fungi at Nadia group.
Cool.
And we found many new viruses. It would be nice to see their viruses, more not phages, because phages are just bacterial viruses. So there is still a huge work to be done on on viruses in general, not only phages.
I could not agree with you more. I mean, the amount of assumptions that had to go into that estimate that I produced hurt my head, honestly. We did a bunch of analysis. So we went into basically all viral sequences that have ever been produced from metagenomes on individual bacterial isolates, and then quantify how many unique species you see there. And so one assumption that has to go into this is that species don't share similar viral species. I'm just going to call it species for the purposes of operationally talking. And we found that that's generally true across super dissimilar bacterial hosts. They generally same. Then we had to say, okay, well, how many do you find out of bacteria? And this is what I wouldn't know about fungi. So we were able to find enough data out there to estimate how many unique viruses you tend to find on an individual bacterial species. And so from there, you could then use bacterial estimates to estimate phage diversity. And you can assume, well, bacteria are the most biodiverse group. I mean, they put back fungi to shame in terms of species numbers. And then we could kind of ignore everything else. But that's not really true, you know what I mean? It's just possible when you're doing a large global inventory. I agree with you. It would be super cool, and I think a super important starting point would be how many are the viruses that are found on individual fungi unique? how many do they share across closely related species, and generally how many unique viruses are found on individual fungal species. This would be a really important thing, I think, to figure out. So I know that also wasn't a question, but I'm also really into that, so. Yeah, yes.
First of all, thank you very much. Great talk.
Thanks.
Really enjoyed it. You showed this correlation between soil carbon stocks and tree growth and tree carbon stocks for the mineral soil.
Yeah.
So my first question is, how did that look like for the organic layer? Because there are data on the organic layer apparently.
Yeah, absolutely.
And B, how deep was the sampling? Because this also matters, of course, because if I'm not surprised that if you just look at the first 10 centimeter in a bottle, for example, compared to a
totally. Yeah, that's a super good question. I had to think a lot about how to try and present those results. For the organic horizon, the correlation is not significant, but it looks really great. So if you don't account for any other covariables, it's negative correlation. But then when you include a bunch of other covariables, it's just not significant anymore. There's a statistical reason why we might ask, like, well, should we include those other covariables? And in general, I would just say the trend is to be negatively correlated, which is fascinating, especially because the one thing I didn't get to show here is that microbial
was negatively correlated.
Yeah.
In Switzerland, it's the opposite, huh?
Yeah, this is just at the European scale.
The larger the tree biomass, the less carbon we have, huh?
Yeah, and like I say, it wasn't significant, so this is a trend that's not significant. So I'm not like saying it matters, just what the observation looked like. Yeah. So it's also interesting to me because the microbiome was super strongly correlated to variation in rise in carbon stocks and in quite unique ways. Oh, and the depth component. So yeah, that was 0 to 10 centimeters I was showing. But if you look at carbon stocks at 0 to 10 and then compared to 0 to 80, the R squared is like 0.8. So it's super strongly correlated. So you can then assume that the entire soil carbon stock in the mineral horizon is positively correlated with tree growth and biomass carbon stocks. Yeah.
You showed us a great picture of species diversity.
Yeah.
Or at least that's what I assume that it is species diversity, and coming more from a within species diversity perspective.
Yeah.
So how much of within species diversity would you expect, or is that just like a conceptual difference that if we look at larger living organisms, we tend to group species at a higher level of integration than you would do that for bacteria or phages or whatever?
It's a really fantastic question that I super don't have an answer to, honestly.
Yes. What would you guess? Is there a lot of diversity within a given OT?
I really can't comment on this for bacteria. For fungi, I can comment on it a lot more. It's complicated for fungi because you have quite a lot of variation within the marker that we specifically focus on. So that variation can be as high as 2%, which is why we tend to try and focus at a 98% similarity for the ITS region and not a finer threshold like you might do for bacteria. So based on the ITS region, like the full ITS region, not just one, we try to be a little bit careful within like accounting for a 2%. For that. That doesn't mean that there is an incredible genotypic diversity that's super important. There definitely is, especially coming from North America, where you have the same species, but they're so geographically separated. They have different names. They can do different things. They respond to nitrogen deposition differently, but they're still 98% similar in the ITS region. So there can be so much diversity at the strain level that we're just not getting at the moment. And it would be nice to be able to do that, but I don't think we yet have a good marker for doing that can link it to taxonomy yet.
Maybe require more than one marker.
Absolutely. I'm thinking we probably need whole genome sequencing for that. Yeah.
Yeah, thanks, Mark. Great talk, and I really appreciate how you advocate for the soil biodiversity. You also push it really far. I mean, that biodiversity really drives also tree growth and everything. And to me, it's more like
I did not say the word drives a single time.
Sorry.
I've learned my lesson to not say that word.
You can call it like this. But possibly it's like to me, it's more like a mutualistic relationship. I mean that tree growth or biodiversity, drives the fungal community, what is below ground, and in turn you also have a kind of a positive stimulation of tree growth. So I mean, I would rather push it like this than really. I mean, maybe the foresters usually forget about the below ground, and this is why it's really cool to push it,
Sure.
But yeah, you have to see both components, I think.
I would argue I did. Honestly, I only pushed on this because I've thought about this so much, and I've also changed to talk about this. Yeah, like for example, don't say drive anymore. We don't know what the causality is there. I try to be pretty frank about that as well. But I agree with you. It's probably, if I had to speculate, part of a series of feedbacks, right? Where like plant growth, allocation of carbon to mycorrhizal fungi is influencing what they do, selecting for certain species that in turn shapes their growth and nutrient uptake. And it's just totally impossible to represent in like a, yeah, I couldn't agree more. I wish it was easier to kind of like get at that. It's just so hard in real ecosystems, you know what I mean? Yeah, but totally, I agree. We have to be careful, and I don't think anybody should be pushing types of like observational correlational analyses as causal. It's pretty dangerous, actually. Yeah.
Hey, great talk. So you spoke at the beginning about dispersal agents, and you explored some of the biotic factors. Have you ever looked at like and broadleaf and conifer trees, and they have very different dispersal agents, not kind of generally.
Yeah.
Is there any analysis that you've done looking at, say, like if trees that have small rodent-based dispersal have more similar communities than those that have, say, like wind-based dispersal?
Ooh, that's a super cool question. No.
Okay.
So I have looked at the relative importance of small mammals versus wind within hardwood stands in New Hampshire. And at the Hubbard Brook Long-Term Ecological Research Site, they've done some really interesting clear cuts there and put a bunch of funnels out to collect spores that would be from the wind, characterize the fungal communities that were there, and then collected a bunch of the primary small mammals that are in that area and characterized their scat. And it was pretty amazing, at least in that ecosystem. The diversity of mycorrhizal fungi and scat is so much higher than it is in the wind, which makes us think that these mycorrhizal fungi's primary form of dispersal in that forest is small mammals and not wind, because in the wind it was way more saprotrophs and pathogens. And this was just for ectomycorrhizal fungi, but we would strongly expect to see something similar for our vascular mycorrhizal fungi too.
Cool.
But answering that question would be so cool.
Yeah, it would be really interesting.
Yeah.
Cool, thank you.
We should maybe talk about that because that would be fun. Yeah.
Hang on. Great talk, really cool. Thanks a lot.
Thanks.
During your talk, it's not my business, but I was wondering during your talk what's the implication of your research on common garden experiments, and how, to what extent, soil microbial communities for structuring or richness can drive tree growth, or disentangle different patterns for different species.
Yeah. So for the common garden experiment stuff, it makes me really nervous when there's no real microbial community, and then we've garnered all of our aspects of plant physiology and plant growth based on a total absence of any microbes you'd ever find in the wild. So I'm really skeptical of that. And that to me makes me think that we need to like wild these types of plant-based studies if we want to get at something that we might expect to observe in a more realistic ecological scenario. I think what those are getting at are more like the genetic potential or the potential with a synthetic microbial community that you might see, because so many of these potting soils are inoculated with specific microbes as well to try and reach a carrying capacity to offset pathogens. So it's just maybe not representative of the extent of variation we would observe in a more realistic ecological situation. So my preference argument would be that people do inoculate them with a microbial community that they could find in a similar type of ecosystem. So if you're studying a grass, maybe you could find an area where that grass natively grows and use microbes from that in your common garden experiment. Yeah. I think there might have been a second part of that question, but I kind of missed it.
We'll have one online just to mix it up.
Yeah, a couple of questions. Yeah. First, Nadia Keller said, thanks for this interesting presentation, Mark. I was surprised by the correlation you found between soil organic carbon and tree growth. Were all sites equally disturbed or primary? I would expect higher tree growth rates in disturbed sites, but I would also expect loss of soil organic carbon because of the disturbance, priming, etc.
Yeah.
Referring to worldwide means,
for forests of Borneo?
Yeah, that is a fantastic question. So it's like in Europe, I don't think any of them are proper primary forests. They're supposed to be representative forests in a forest inventory. So the vast majority of them, / maybe all of them, are not primary forests. So you can think about this as really not being applicable to a primary forest. And that kind of gets at what you might observe in a primary forest. No disturbance, perhaps a lot of carbon is accumulated, and you would no longer see this relationship, or the relationship would be saturating, or perhaps even negative if some type of disturbance were to come in. Totally agree with that. I think the reason is because they're not primary forests. And there's an incredible range to the extent of human management, because we're talking about all of Europe. So some of them are no longer really managed at all, and some of them are more intensively managed. It just depends on the country. And even within the country, it depends on the stand.
Okay. And a short question. Yuan asked, for the study about the similarity and dissimilarity of fungal community or bacterial community in the European forest, why clay is considered as an important factor?
Sorry, what was the
Clay.
why?
Why is
Clay.
clay considered important?
Yeah, okay. That's important because it's a significant proportion of the substrate that organisms are living in. And experimental work has shown that when you manipulate clay and sand content and you put it out into the environment, you can select for different microbial communities. And so we found for both fungi and bacteria, again, this isn't relevant in the organic horizon, but in the mineral soil, it emerges as an important factor that it captures variation. Yeah. There's a lot more reasons for that than just what I said, but that's one of them. Yeah.
Yeah, I just wanted to come back to Felix's question, kind of. I was wondering, why do you actually have to go over species classification and OTUs? Couldn't you just use the sequences that you get and calculate diversity and differentiation in a population genetic, population genomic way instead of going through counting species?
Such a good question. So that is something we did for bacteria. That would be the ASV approach, so amplicon sequence variant approach, which is that. That's where it doubled the estimate of species that we had. But for fungi, that approach isn't a good approach because there's well-known intragenomic variation in that marker. And so if you counted them as being distinct based on just the exact sequence variant, you'd be totally overestimating fungal diversity.
Okay.
But you will see a bunch of papers that do that incorrectly because it's really easy to make ASVs and it's way harder to make OTUs. So there are a lot of fungal papers out there that do produce ESVs, ASVs, so just a single sequence. But that's something we have to be a little bit skeptical of. Even if you might see the same patterns, the levels of alpha diversity you'd see would be inflated. Frank?
Yeah, maybe I have another question to ECM. Also towards carbon sequestration, because it may also play the opposite role. I mean, that ECM may, of course, stimulate tree growth because it helps the tree to get their nutrients.
Yeah.
But at the same time, the ECM also mines organic matter to get to the nutrients. And by this, they are decomposing organic matter. And in Sweden, you actually observe a pattern that the more ECM you have, the less soil carbon you have.
Yeah.
So, yeah.
Yeah, absolutely. That's why that correlation between tree growth and soil carbon is not inherently what we would expect because that's important. And ectomycorrhizal fungi, if we look at all the sequences and if that's at all a proxy for the proportion of the community, are dominant next to saprotrophs. So I think it's going to depend a lot on what we see there. I think it's going to depend a ton on substrate input. It's interesting what you say about Sweden because the prevailing narrative at the moment is that in forests with a lot of lignin input, with a high CN ratio, I'm guessing you probably are talking about pine forest, you expect to see gadule effect, but that's just not consistently observed. And so I think we need to question the underlying theory behind the gadule effect a little bit more, especially even modern takes on it that have been presented recently. It's not the case. And I also found that when I looked at some of the indicator species in the organic horizon of low carbon content, there are species we expect to be strong decomposers. A lot of species of Cortinarius that we didn't find in the organic horizons with a lot of carbon, which again goes totally against the gadule effect. So I do expect in a lot of ecosystems, some ectomycorrhizal fungi are going to reduce primarily in the organic horizon carbon stocks. I think in the mineral horizon, we might see something different because their biomass that's generated also constitutes a significant amount of necromass that could cause accumulation in the mineral horizon. But that's the next level of complication to the Gadgill effect that hasn't been explored.
There's time for one more question.
Do we know how the bacterial biodiversity is structured into the soil? So from the root tips or from the root system to the soil through the rhizosphere? Do we have that? Do we know anything about, or to the depth, or how? Do we have any clue about that? Or?
I don't. I'm sure other people do, but like I'm just masquerading as a person who knows anything about bacteria. So I don't know very well there. I don't know, maybe you'd have a better sense of this than me. No, okay. Or Frank, maybe you would. No, okay.
So this 9 billion of species are more or less homogeneous through the soil. Through the soil.
Ah, sorry, that's what you're asking. Yeah, okay. So like, do you expect to see higher diversity levels in specific areas, kind of?
Yeah.
Yeah, so
Next to the roots, or what? How would you guess?
There is research on that.
And for sampling is important.
Definitely agree. I don't remember off the top of my head if rhizosphere soil is more diverse than bulk soil. You often see that in sequencing campaigns, that the rhizosphere soil is more diverse than the bulk soil, but the bulk soil is often more uneven as well, so it's harder to get at the full diversity. So I'm not sure I would be confident to answer that question. I can definitely tell you the organic raisin is way more diverse than the mineral soil, but that's about it.
Okay.
Yeah, good question though. Cool. Thanks everybody online.
Thank you.